So let's suppose you wanted to make a vaccine. How would you do that? I think the first thing is to try and see, what diseases out there are hurting children or adults. And once you then find what that disease is and find either the virus or bacteria or fungus or parasite that's causing that disease, then what you have to do is try and figure out, okay, how can I make a vaccine? Now, actually, all vaccines are made the same way, which is you're trying to sort of separate that part of the microorganism that makes you sick from the part of the microorganism that induces an immune response which protects you. That's the way every single vaccine is made. So, for example, when Jonas Salk made his famous polio vaccine, what he did was he took polio virus, he completely killed that virus with a chemical, but he didn't affect the virus so much that when you injected it into people's arms, that it couldn't induce an immune response. It still induced an immune response, but because it couldn't reproduce itself, it couldn't grow, it couldn't cause disease. And that's how he made his vaccine, his so-called inactivated polio vaccine. And that's how you make any vaccine. You're just trying to separate those two things out. Sometimes you just weaken a virus by a number of means so that it sort of reproduces itself a little bit, not so much that it causes disease, but enough that it induces an immune response. For bacteria, sometimes you just take the surface of the bacteria, the so-called complex sugar or polysaccharide. Sometimes you just use a single viral protein or a killed virus. So there's a lot of different ways to make a vaccine. So the first thing is to sort of identify the pathogen or organism and then figure out where to go from there. But so it goes into several stages. The first is hopefully you can have a small animal model to study the disease. That's not always true, but if you can get it, it makes things a lot faster. But step one or phase one is you have something, you think you've made a vaccine. So let's suppose you were interested in making a respiratory syncytial virus vaccine. This is a virus you may not have heard of, but it's a virus that really causes tens of thousands of children to be hospitalized every year. It kills thousands of children, and there's not a vaccine for it. It's called respiratory syncytial virus. So now you think you've made a vaccine. So the first thing that you do is so-called phase one trials, where you inoculate it initially into adults, and then older children, then younger children, then young children, just to make sure it's safe and to get a sense that you can induce an immune response that may hopefully be protective. Then you go to phase two studies, which is the same sort of things. Now it's not only dose, but it's sort of a bigger safety trial to make sure that it's safe and that you've got the dose right. Then if that all looks good, you can go to so-called a phase 3 trial, which is a much bigger trial. Now you're looking not only to make sure that it's safe, not only to make sure that the dose is right, but also to make sure that this vaccine protects against the disease that you're trying to protect against. And then after you've done that, you submit all that information to the Food and Drug Administration or FDA for approval. Technically, licensure, if everything looks great, takes about 10 months. But the process, as a general rule, the time it takes to make a vaccine here in the 21st century is around 25 years.